Okay, everyone, welcome to the sliding filament theory. This is probably one of the coolest things and theories and descriptions of how we move because it answers exactly that. What is happening in our muscles that allow us to move our bodies? Well, here it is for you. You can wonder no longer by the end of this lecture. Okay, so the sliding filament theory was something that was generated or proposed in the 1950s. And as I mentioned, it explains exactly why and how we have these proteins in our skeletal muscle cells that can contract and therefore generate a force and move our bodies. So, as I mentioned last lecture, the primary gist of how sliding filament theory works, or how our muscles produce force, is we have these strands of protein set up in this alternating structure within striated muscle, and they contract by sliding being pulled across one another. Okay. When essentially, to describe how much force is generated in a muscle cell, ultimately that comes from how many of these little, as I mentioned in the last lecture, like rowers or paddles, or as we'll find out, the myosin heads attaching to actin, however many those are and how much gets recruited across the entire muscle will dictate how much force gets generated. Okay. Well, let's look at this process. We're going to look at it in two different ways. So the first one will be a little bit verbally, step by step. Then we're going to take a look at a diagram. There's also going to be a video posted that kind of explains and illustrates in real time the actions that are happening. I find that quite helpful to see with this complicated process, or what seems initially as complicated. What you need to keep in mind, or something you should keep in mind, is that you will need to know and recall the order of all the events, how they occur, and definitely be able to explain how the two, the actin and myosin, interact to be able to produce force. Okay. So, without further ado, what we'll see is we have a few steps that occur, and we'll start right from the signal of the brain. So our brain sends out a signal, it travels down our spine towards the nerve that intervenes with our muscle. And that's where we kind of start with this process. So we'll look at that electrical impulse that comes from the nerve and goes to something called the neuromuscular junction. And all that is the component where the end of the axon of the neuron intervenes with the muscle. Okay. What you'll find is that action potential, or the signal that travels down that nerve, will actually get transferred to the muscle cell itself. So it's no longer an electrical signal in a neuron, it's an electrical signal in the muscle cell that travels along what's called the sarcolemma. Okay, or you can think of it as like the outer body of the sarcomere. Okay. As that signal travels along, it ends up finding these little, what's called transverse tubules, or T-tubules, and the signal can travel down deeper into the muscle itself. Okay. And what will happen is those T-tubules, you can think of the little tunnels into the muscle, reach this holding component of calcium. Okay, it's this big storage system for calcium, and that's called a sarcoplasmic reticulum. Okay, or SR for short, but sarcoplasmic reticulum. Okay, it reticulates or retains calcium. Okay. Again, whenever you hear the word sarco, you can think of muscle. Okay, sarcolemma, sarcomere, we know from last day is the basic unit of a skeletal muscle cell. And now we have sarcoplasmic reticulum, this holder of calcium. Okay, So we got this big storage unit of calcium that now has just had a action potential or a electrical signal reach. And what happens is that electrical signal causes this holding vessel to let out all of its a lot of calcium into the sarcomere or sarcoplasm of the muscle fiber itself. Okay, so now we're at the muscle fiber, we're at the actual proteins finally. All of that was getting there. Okay, so quite a journey so far. So we're at the muscle fiber. We've now had our sarcoplasmic reticulum release this calcium, and what happens from there is the calcium goes and finds those little binding sites on troponin. So if you remember, troponin had three subunits, right? One that attached to actin, so the thin filament, one that attaches to troponin, which is the wire around actin that it holds onto, tropomyosin, and then the third subunit of troponin allows calcium to bind to it, and it has a high what's called affinity, so it really likes binding to that site. Now, this is where the fun starts really happening. So once calcium binds to this little subunit, things start to change shape. So if you remember, structure is function. And as we've discovered with like cofactors and other things that bind to enzymes that we've spoken about in previous sections, is whenever we have this protein structure, something binds to it, it changes the shape. And sure enough, that's exactly what happens here. Once calcium binds to these subunits, it changes the shape of the subunit and pulls back tropomyosin away from binding sites on actin. Okay, so again, we have this thin filament, actin, which was getting guarded or protected by tropomyosin. Okay, troponin had calcium bind to it, and all of a sudden it's moved tropomyosin away from actin. Okay, now what's the big deal about that? This is like a really big step, okay? What will happen here is the myosin, so now we're talking about the thick filament with those globular heads or the big paddles, right? They can now attach to actin. So again, when tropomyosin moved back, what can happen is it has little active sites now where myosin can bind to actin. Okay, so finally now, after all this action potential, the signal that travels along the sarcoplasma or sarcolemma down the T-tubules into sarcoplasmic reticulum, releases calcium, which then binds to the troponin subunit, which moves tropomyosin, which allows actin or active sites on actin. Remember, actin is active, has active sites. The myosin binds to it. So we finally have our thick and thin filaments connecting. Okay. Now that we have that, what we see is that's where the actual moving and contraction and power or force production occurs. Okay, and that's from what's called cross-bridging ratcheting, and we'll get to that ratchet action in a bit more detail. But for now, you can just imagine that cross bridge happening. The myosin binds to actin, pulls. So again, myosin pulls actin towards the M line or the center line of the sarcomere. And then that's when we get force produced. Okay. What happens to kind of reset everything is the sarcoplasmic reticulum starts sequestering or pulling back all the calcium that was released into the sarcomere or into the cytosol of the muscle fiber. So sequestering it all back, pulling it all back, and as those calciums leave the troponin subunit, or troponin, so it leaves troponin, what will then happen is tropomyosin covers up or blocks out all the active sites on actin, the thin filament, so myosin can't attach to it anymore. Okay, and now we're reset. Okay, again, quite a journey, but absolutely amazing how this process is exactly what causes our muscles to contract and therefore allow us to move, allow us to speak. And you can just think of how frequently this signal occurs, and then again, like calcium gets sequestered and fired again. The signal arrives to our muscle and then taken back to allow us to move as quickly as we do, to allow us to do things like even speak, move our mouths in such a dexterous way. So pretty amazing stuff. All right, that was the verbal version. Let's take a little brief overview. And again, this is good to revisit multiple times and really order the process in your mind. So let's do that a little bit with pictures now. Again, just to really reinforce that. So if we have our nerve here, and I'll pause briefly before we do this. I really want to encourage all of you to get a blank piece of paper, get a whiteboard, use your tablets, whatever, and draw out this entire process step to step, labeling each part, making sure everything occurs in the right order. It really helps solidify things. The more you can just fill in that diagram and that process from memory, the better off you'll be come exam time. Okay, that's my sales pitch and how to go about my tip for how to study this. So again, we'll start off with the signal coming down the last neuron that's reaching our muscle. So we have our neuromuscular junction, which is right here. So we have the synaptic cleft, which is the space between a nerve or neuron and the other side of whatever that happens to be. Usually, it can be another nerve, or here we have a muscle. From there, once we have that action potential from acetylcholine being released into that synaptic cleft, we have acetylcholine released. It's really important, and causes that action potential to travel down along the sarcolemma into the T-tubule. Okay, so you can see that it's called transverse tubule, T-tubule. That action potential continues down to the point where it reaches the sarcoplasmic reticulum, which is this blue stuff here. So again, that's the bag or the components or sheath, the covering of these muscle fibers here that hold calcium. Okay, we have the signal that travels down, tells sarcoplasmic reticulum, hey, dump your calcium. Sure enough, those voltage gated channels, so right here, so again, voltage gated means electrical signal comes and tells them to open. So they do, and they release from that depolarization all the calcium towards troponin, and therefore moving tropomyosin out of the way off of our thin filament actin here. Once that occurs, myosin can attach the globular heads to the active sites on actin. Okay. Once that happens, we get a cross-bridge contraction, and we're going to go through the specifics of that shortly. So you can just think of it as the pulling or the power stroke or the power production of the shortening of the muscle. And then again, to finalize things, say return to resting state, the sarcoplasmic reticulum takes back or pumps back into it the calcium, so that there's no more here that's available to bind to troponin. Therefore, tropomyosin covers up and blocks myosin from being able to attach to actin. Okay, quite a few new terms in there. Some are very similar, but different enough to necessitate their differentiation. Okay, so that's the overall process, but I kept leaving this little black box, this little crucial component of how muscles contract and generate force. I kept skipping over that part a little bit and saying we're going to get to it later. So let's get to it now. No time like the present. So again, just to put it into context, we'll go through the steps. This does still occur, action potential from our nerve down through the T-tubule, causing calcium release from the sarcoplasmic reticulum. That's all happening, but we're just going to focus on this action here, or the interaction between actin and myosin. Again, they are the contractile proteins of our skeletal muscle. So let's focus on that for the next component. Okay. How does this work? And I think more importantly, what's great about this cross-bridge contraction model is it helps us understand why and when and how are we actually using ATP, right? We spent much of the semester talking about our different energy systems that produce ATP that allow our muscles to contract. How is that being used? When is it being used? And how does that move our body? How does that cause our muscles to move? Okay, well, here it is. This is the thing to know. Definitely know this. So we'll start off on the first step, and it is really helpful, I find, to think of this as a cycle. So we'll start off with our binding sites, or our active sites on actin being exposed. Okay, so what's happened so far? Well, we have calcium released, right, from our sarcoplasmic reticulum as a result of those voltage gate calcium channels being opened. And we see that we have some ADP and phosphate still on our myosin here. So this magenta type purple is our myosin, and our lavender purple is our actin. Okay, our troponin and tropomyosin are the darker purples here, up close to our actin, right? Okay, now that we have the stage set, let's again see how this goes. So we have these active sites, which are kind of the pink spots on the actin, open and available for myosin to bind to them, okay? And myosin at this point in time is in a cocked or set position. It's a ready position. And so what happens is once those active sites are exposed, this active, charged, ready to go myosin will attach. So we see this cross bridge moment where cross bridging occurs, myosin attaches to actin. Then what will happen is we get the actual movement. So there's physical movement where these myosin cross bridges pull the actin. So this is called the power stroke. So it pulls the actin towards the M line, which will pull the Z discs closer to one another. Okay, just a little bit, because we just have a few globular heads here doing that. As the power stroke occurs, the ADP and phosphate group get disengaged from the myosin. Okay, so they kind of fall off or get released, and that's good. That's important, because our next step is a new ATP binds to the myosin. And what happens at that is it releases from the actin. Okay, so if there is no ATP present at this point in time, and we just had ADP and phosphate, the myosin actin cross bridge would stay in that pulled non-reset state. So it would stay there. Okay, but again, we get ATP that comes in, and it causes a release. Okay, so it releases from the actin. Okay, what's important about getting a new ATP on the myosin head? Well, when it becomes hydrolyzed, or when we release the energy from ATP to be ADP and phosphate again, that energy release energizes the myosin into an active position, or active state, or active conformation. Okay, so then we say that the myosin head has been cocked or set, or ready, essentially reset to be able to find another active spot, attach, pull, or power stroke release ADP and phosphate, get re-energized, let go. ATP gets hydrolyzed or releases energy, resets the myosin head. Okay. So really important to know these steps. Some models or versions out there will put this into 4 steps. I quite like having 5 steps because it really puts emphasis on the ATP attaching to cause release, but then that separate step where the ATP gets hydrolyzed to create new energy and it needs to have those active sites on actin, otherwise the myosin can't bind to it. Okay, so that's really important. Alternatively, step 5, if calcium has been sequestered back into the sarcoplasmic reticulum or pumped back in through calcium pumps, what will happen is, again, tropomyosin will cover those active sites, and we won't have another cycle ready to set. It'll just wait in the energized state, ready to contract again. Okay, so again, this is pretty wild stuff considering how this simple, relatively straightforward process where we have these little globular heads attaching and pulling, detach, attach, pull, just over and over and over. That's what causes our muscles to move. And again, so this is one little microscopic component of one sarcomere. Each sarcomere has lots of globular heads. They have lots of thick and thin filaments. That's one sarcomere. You expand that or multiply that across an entire muscle fiber. You have muscle fibers that are redundant in multiples throughout each muscle. So you can see how, again, just by sheer vast volume of numbers of protein fibers in the body, that's how we generate force and move our bodies with our skeletal muscle. Pretty spectacular, hey? I think this is wild. This is absolutely amazing stuff. And for sure, again, let me say it again, you will need to know this entire process as well as this, okay? They go hand in hand. So from the start of the action potential coming down our motor neuron, all the way to the point where you can complete this cycle of contraction and explain when it ends, which is what we'll speak about now. So there's 2 ways we can get our muscle back to a resting state. And again, if you recall, one of the most important things is that removal of calcium. So once that happens, the muscles will typically stay at whatever length they are at. So how does it return back to a resting state, ready to go again? Well, one, some sort of outside force has to work on it. So typically, we'll think of that as gravity, although it could be someone else passively moving us. That's potential. But a lot of the times it's the work of an antagonist muscle. Now, we haven't covered antagonist and agonist muscles yet. And I'll just use in brief the example of a bicep curl to help explain that. So we have 2 muscle groups that oppose each other. So in one case, we have something, say, our bicep. On a bicep contract, my bicep shortens. What will happen is the tricep will relax or shut off. So again, that's the bicep becomes the outside force that moves the length of my tricep to get longer because it's actively not contracting, it's not getting action potentials, it's not going through any cross bridging. The tropomyosin has covered the actin. So again, the myosin doesn't want to attach to it. So we can lengthen that muscle. Okay. The deltoid might be active, and I like bringing this because it shows how the complexity of movement can be. So the deltoid might contract and hold isometrically. So that means it will be in a set state. But that contraction, because there's no movement, it's just considered a stabilizer. So it's not an agonist, not an antagonist, even though it might be involved in stabilizing the limb. Okay, so we have 3 kind of different contractions: isometric, concentric, and eccentric. The agonist will contract, the antagonist will relax, and these work in opposition. And you can try this yourself. If you try to flex both your bicep and your tricep in a co-contraction, your elbow won't move in either direction. So that becomes an isometric contraction. But if you relax your tricep and just bend your bicep, tricep, if you poke at it, should feel nice and supple, kind of relaxed. Bicep should be hard and contracted. Okay. All right, and that's how we return a muscle back to resting length. All right. So as a recap, definitely know all of sliding filament theory. Again, it's this beautiful theory that explains how we move our muscles and move our bodies. Cheers.